Well, in the processes of biotechnology, we had primarily studied how do we separate out the sort of a DNA. After we separate out the DNA, it is our primary goal to separate and isolate our DNA fragments. As implied, we had the sample DNA. Your sample DNA could have the gene of interest somewhere. Then again we have got the plasmid DNA. The plasmid DNA is present in the cloning vector, and the plasmid DNA and your gene of interest and the entire sort of DNA are cut by a common core of restriction enzymes. We send in restriction enzymes which will chip the DNA at different places, and it will also chip your sort of a plasmid. Thereby you get small pieces of the DNA on either side. Once we get the small pieces of DNA on either side, we need to separate the DNA fragments. We need to separate the DNA fragments, and the technique by means of which the separation is achieved is termed as gel electrophoresis. Electrophoresis is a sort of a technique wherein by using an electrical field you can separate biological molecules. You separate biological molecules because biological molecules are amphoteric. They have got a positive and a negative charge, and when you send in the sort of a molecules on a sort of a migration, that is, you separate them or you analyze them, they get separated into different sort of bands based on their molecular weight and based on a migration in a sort of an electric field. So here we would be explaining, now I would be explaining primarily about the entire technique of gel electrophoresis. So gel electrophoresis is a technique of separation of the DNA fragments based on their size, and sample DNA fragments of sample DNA are cut into smaller fragments by restriction endonucleases. Then the DNA fragments are charge separated. The DNA fragments are charge separated when they are loaded into a gel column. When they are loaded into a gel column. So here the most important, the basic technique of gel electrophoresis is as follows. The gel electrophoresis consists of a glass plate on which a gel made up of agarose, which is derived from an algae, is added. The gel is put just like a sort of a wet cream, and when the gel is put, it is fully loaded and it is left overnight. After it is left overnight, welds are cut in the gel column. Welds are cut in the gel column. The entire gel apparatus is connected to a sort of a electric current. Once it is connected to an electric current, the agarose, which is a sort of a polymer, and it is made up of galactose. It cross links and goes to form the sort of a matrix. Now, whenever we have DNA, now you take the sort of a DNA, and you load it into the sort of a well. You just put a few drops of the DNA into the sort of a well. You can load it by means of a sort of a cuvette. By a cuvette, you load the sort of DNA into the sort of wells. Now, depending upon their size, the agar acts like a sort of a sieve or a sort of a gel, and we have the migration of the DNA molecules. Down the electric field. We have the migration of the DNA molecules. Now, the larger molecules, supposing we have the larger molecules stay on the top, whereas the smaller molecules move at the bottom. These are the smaller molecules which are having low molecular weight. They migrate faster, whereas these are the larger molecules which stay at the top. So, all the DNA molecules primarily they migrate, and when they migrate it moves in a particular sort of a column. So, this column is termed as lane. It is termed as a lane. So, they move primarily in the case of lane. Now, I have just drawn a sort of a typical sort of a gel electrophoresis apparatus. We have the anode over here, we have the cathode over here, and your DNA is loaded into wells. Supposing this well 0, this well 1, this well 2 and well 3. Lane 1 is called control, into which nothing is loaded. Then you have your DNA element, the DNA just swiped and put into the sort of a well. When the electric current passes, the DNA will separate into different sort of a bands based on its molecular weight, based on its size, based on the sieving effect, based on the charge. Based on the size, the sieving effect and the charge, the DNA molecules get separated into different sort of a bands. So, the bands get arranged on typical lanes. Vertical columns of the gel are termed as lanes. So, you got lane 1, 2 and 3. Once you have the typical migration of these particular lanes, has implied the heavier DNA, the heavier of the DNA, the heavy DNA bands always tend to be at the top, and the lighter ones, the light of the DNA of the lighter molecule, lighter DNA bands appear at the bottom because their molecular weight is less. So, what we have done is we have taken DNA and we have subjected them to a restriction digestion by enzymes. Then we have subjected them to gel electrophoresis. So, this gel electrophoresis is termed as polyacrylamide gel electrophoresis, or it is popularly called as PAGE. So, here it has been subjected to gel electrophoresis. Then we understand that DNA can migrate. It migrates in the form of bands. After it migrates, we need to visualize it. We need to exactly know what is the sort of a DNA, or where exactly is your sort of a recombinant DNA. Now, to visualize it, we need to stain it. So, normally what would happen is when you are loading your sort of a DNA, we also add a dye which is termed as ethidium bromide to the sort of a DNA. We add a dye called ethidium bromide. So, the ethidium bromide dye, primarily when you get the bands, after you get the bands, you go to primarily remove a particular sort of a lane. Supposing this is the gel column, you are using a chisel or a knife or a blade, and you're cutting the gel column. Once you cut the gel column, you have the different bands of the sort of a DNA over here. So, you need to cut this band. So, you are cutting the column, and you are extracting your sort of a DNA. You are taking the sort of a DNA. So, you would typically have a tray in which you would have the gel, and you would have cut the sort of a DNA like this. And when you cut the DNA is already stained by means of ethidium bromide. So, when you extract the band of the DNA, you cut the gel column, and this process of extracting the DNA by cutting is termed as elution. So, elution is removing the band of the DNA which is strained by means of ethidium bromide in the last stages. So, after you elute it, you need to observe it under UV light. So, you will be under UV lamp, you will just be observing your sort of a DNA. And the DNA primarily, because it is stained by ethidium bromide, it will show a sort of a fluorescence of, it will show orange fluorescence. Thereby, it will have a bright orange color. So, the entire process of extracting the band of the DNA, cutting of the gel column and extracting the DNA is termed as elution. So, after the DNA has been eluted, E-L-U-T-E-D, eluted, that is, after you go through all this visual observation, you remove the sort of a DNA, then the DNA need to be purified. Once it is purified, it is primarily ready for amplification, which occurs in the sort of a polymerase chain reaction. That is, you get your sort of gene of interest, and once you get a sort of gene of interest, you can do selectively, you can from your gene of interest, you can make millions of copies of those particular sort of a gene. Thereby now primarily, as if you just look back, we had a sort of a, we were interested in primarily into plant biotechnology. We had the basic tools are restriction enzymes and cloning vectors. After you have the sort of a cloning vectors, you need to exactly know how to separate your sort of a DNA. After you separate your DNA, you require to have a sort of a competent host. And again, once you load it into the sort of competent host, once you get a gene of interest, the gene of interest need to be amplified by means of the sort of a PCR technique. So, gel electrophoresis also plays a very important role with relation to isolation of the DNA fragments. When you want to isolate the DNA fragments, it is primarily based on the technique of gel electrophoresis.